I'll give you a brief, simplified explanation of tissue-specific gene expression. All the cells in our body have exactly the same gene. For example, muscle cells and neurons in our bodies have exactly the same genes. However, muscle cells and neurons look different. Why is that? Because gene expression is different in different kinds of cells. In muscle cells, certain types of genes are expressed. For example, in this particular case, this gene is expressed, but not these. While neurons express different sets of genes. In this case, this gene is expressed, but not these two genes. So what controls such a difference? The proteins that control the gene expression levels are called transcription factors. The transcription factors are master regulators of gene expression, which bind to the promoters of the genes and activate the transcription of the genes to messenger RNAs. In muscle cells, for example, a transcription factor called MyoD binds to the promoters of particular genes. In neurons, for example, a transcription factor called neurogenin binds to the promoters of some other genes. Then that's why you can get the expression of different genes in different tissues. One way to use GFP is to design a GFP gene that is controlled by this tissue-specific promoter and introduce such a gene into animals. So for example, using this particular promoter, you can replace the protein coding part of the gene with GFP gene. So now, if you introduce this gene into the animal, you can label only neurons by GFP. So you can visualize certain tissues during the development. If you use RFP together with the muscle-specific promoter, like this, you can replace this protein coding gene with RFP, the red fluorescent protein. You can see the muscle cells as red. If you introduce these two together into the animal, you can see muscle cells as red and neurons as green. Another way to use GFP is reconstituted fluorescent proteins, or GFP. As I described earlier, GFP has 238 amino acids. The GFP can be cut half. For example, 1 through 157 and 158 through 238. So these half GFP are not fluorescent. In this example, these two split GFP proteins are fused with another two proteins, NZ and CZ. NZ and CZ. So these two proteins interact with each other. So when NZ and CZ binds to each other, GFP is deconstituted and become the fluorescent. By observing fluorescence, you can monitor the protein-protein interaction. You can also mark particular cells. This is a figure of the real research paper published by Professor Chalfie and his co-workers. When they placed a GFP gene under control of universal promoter UNC24, GFP gene under control of UNC24, which is a universal promoter, and introduced such a gene into nematodes or worm, they observed fluorescent signals from many adult cells. But next, they placed NZ-GFP gene under control of UNC24, which is a universal promoter, and CZ-GFP under control of EGL44, a promoter specific for touch receptor neurons. When they introduced these two genes into nematode, they observed fluorescent signals only from 6 touch neurons. In the next video clip, I will show you more about how to use fluorescent proteins with different colors.